record. All right. Continuing on with genetics and inheritance, and in particular, we're going to move away from Mendelian inheritance. But I'd also like to look at probabilities, because you don't always have the luxury of a Punnett square, for one thing. And of course, we'll lead on from genetics and inheritance to evolution. So when you consider a microevolution is the change in allele frequencies from one generation to the next. And we've been talking about how alleles are inherited. And the probability of you inheriting your particular chromosomes from your parents was 1 in 64 trillion. So that means that you and I, all of us, we are recombinants. So we're a recombination of chromosomes. And recombination occurs in several ways as a result of meiosis. So I'm just touching on meiosis very quickly now, and we'll go over it in more detail later on. So there's two pictures here. One is mitosis. Mitosis is different. Mitosis is when your cells divide. When any cell divides, it goes through mitosis. So in order to divide into 2 new cells, the new cells need to have a full complement of DNA. So the DNA is replicated, and each new cell gets a whole complement, a whole set. So in this case, the chromosomes are here. These are 2 pairs of homologous chromosomes. This is one pair here, and this is another pair here. Homologous chromosomes. You know what? I should have done that in a different color. And remember, homologous chromosomes, I should write this down. Homologous chromosomes code for the same genes, characteristics. But this is very important. It's so important, I'm underlining it 3 times. But they may be different alleles, different versions of that characteristic. So in replication, in mitosis, what you have are exact duplicates of every chromosome. And those are called sister chromatids. Exact replicates or duplicates. And then while the cell is going through mitosis, they all line up in the middle. And that is called metaphase. And then the chromosomes, the sisters, get pulled to opposite poles of the cell before the cell divides into 2. That's called anaphase. And then the cell goes through cytokinesis or telophase, and you get 2 daughter cells. But this is the kicker. The daughter cells are both exactly the same as the parent cell up here. But meiosis is different. So in this case, we started off with 4 chromosomes, and our daughter cells each have 4 chromosomes. That's perfectly normal. Sorry, it's so messy. It is quite messy. In meiosis, you're kind of going through 2 mitosises. You're kind of going through 2 mitosises. So we're starting off with the same chromosomes. So here's one pair of homologous chromosomes. And here's another pair. And then they duplicate just the same as they do in mitosis, but they line up differently. So in this case, they all lined up in a row, but over here, the homologous chromosomes lined up beside each other. So this is one homologous chromosome with 2 sisters, and this is another homologous chromosome with 2 sisters. And then the homologous chromosomes in the first division get pulled to opposite sides of the cell, and then, and only then, do the sister chromatids get pulled apart to opposite sides of the cell. So when the sister chromatids get separated in the daughter cells, we're left with not 4 anymore, but 2 in this case. So 2, 2, 2, and 2. Each cell has 2. And that's really important. That's really important. So in meiosis occurs with our germ cells. So with all organisms, meiosis cuts the number of chromosomes into 2. So cuts them in half would be a better way of saying that. Yeah. So what is so important about that? Can you imagine if that didn't happen? So say, your parents gave you each 46 chromosomes each. That would make 92. And then you passed all those on to your offspring, and they would have 184. And of course, the number of chromosomes would build up from generation to generation, which is ridiculous, of course. So instead, the chromosome numbers are split in half, and you only get half from each parent. And the way that occurs is through the process known as meiosis. And of course, the chromosomes hold the genes. So let's look at probability. What happens during meiosis to homologous chromosomes? Well, they segregate, and they segregate independently of each other. So that means that, I'm just going to try to go to one back. That means that they segregate. Yeah, we know that. They segregate to opposite poles of the cell. And we also know that these chromosomes here separate independently of these ones. So the chromosomes are independent of each other. In the terms of probability, the probability that 2 or more independent events occur together is the product of their individual probabilities. So if you tossed a coin once, a nickel, gosh, I haven't seen a nickel in years. The probability of getting heads is 50%. But what about getting heads twice in a row? Well, if you get both twice in a row, well, you multiply them together because that is much lower of a probability. And it applies to our monohybrid crosses that we did before. So in this case, this diagram shows a coin being tossed, and there's a 50% chance of getting one, 50% chance of getting the other, heads or tails. We use letters, but the principle is the same. Same for the eggs. And then to get one and the other, we need to multiply those probabilities together. So I'm telling you this now because later on we're going to be looking at the evolution of populations. So in a population, the occurrence of alleles can be different than 50% when we're talking about the whole population. So even in the population of our classroom, we don't have exactly 50% of us have blue alleles and 50% of us have brown alleles. No, no. It might be quite different. But the principle is the same, probabilities. So we multiply 0.5 times 0.5, and we get 0.25, the probability of getting that particular one.
Is
Now
that
we also, sorry?
Oh, sorry, I just had a question about that.
Yeah.
Within the population, is that why in a certain population you would have like more prevalence of diabetes or like
Yes.
illness like that?
Yes, absolutely. Yeah. More prevalence of one character over another character is definitely due to the uh, um, variation in alleles in a population. Yeah, that's a good question. So the addition rule in the language of probabilities is that any one of two or more mutually exclusive events will occur as calculated by adding them together. And it's good to look at tossing a coin here because if you toss a nickel, there's 50% chance of getting heads, of course. If you toss it once, there's a 50% chance of getting tails. So what is the probability of getting heads or tails, one or the other? Well, of course, it's 100% of getting one or the other. So we can apply the rules of probability to predict the outcomes of crosses involving multiple characters. So it gets a little bit more complicated with dihybrid crosses, but a dihybrid or any other multi-character cross is equivalent to two or more independent monohybrid crosses occurring simultaneously. So if that were to occur, you can calculate the chances for various genotypes. You just consider each character first, separately, and then the individual probabilities are multiplied together. So if you need to do that for a dihybrid cross. And we're not going beyond dihybrid crosses in this class. So that concludes probabilities. I hope that made sense. And it's handy to have probabilities in order to try and predict what characters will be like in a subsequent generation. So when you're doing pedigree analyses or other genetics analyses where you're trying to predict what individuals will inherit in the future, then it's good to know what the probabilities are of inheritance. But not everything is Mendelian. There's loads of non-Mendelian inheritance patterns. They're sometimes quite complex. So, for example, a single gene, well, it can have complete dominance. That's Mendelian. But it could also be co-dominant with another allele, and there can be incomplete dominance between alleles. Some traits with dominant inheritance, Here's some traits. Having mid-digital hair, that's a dominant trait. Bent little finger, dominant. A short big toe where the second toe is longer than the other ones, that's a dominant trait. A white forelock we've looked at, we talked about a little bit before. It's dominant inheritance, interestingly. Sometimes it is associated with other problems like hearing problems. But the thing about dominant traits, if you're looking at a pedigree, so this is a pedigree, you will notice that they don't skip generations. So at this point, I'm going to explain what a pedigree is. You might have heard of pedigrees before. Usually pedigrees are associated with dogs or horses, but humans, of course, have pedigrees as well. And this is a multi-generational pedigree. So these, I guess, are the great-grandparents, grandparents, parents, offspring. So a box is male. The circle is female. A filled in box or circle means the individual expresses the trait. So all of these individuals express the trait. This female, 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 male. Yeah. So brachydactyly, that's the shortening of fingers and toes. That's also dominant inheritance. And here is an incidence of, I think this is a royal family. So there's loads and loads of data. But you can see again a couple of things. One is that the trait does not skip generations. Does that mean it's dominant or recessive? It's much more
Dominant.
likely to be dominant. Another point by looking at this pedigree is that the squares and the circles are both filled in, which means that males and females both exhibit this trait. Because sometimes we're asking the question, is this trait an X-linked trait or a Y-linked trait, or is it an autosomal linked trait? So a trait that's not associated with the sex chromosomes. Achondroplastic dwarfism is also an autosomal dominant trait. So it's not, autosomal means non-sex linked. And there can be some other issues involved as well. Sometimes abortions and stillbirths seem to be of greater frequency. Homozygosity for this allele could be incompatible with life. Porphyria, that's another one of dominant inheritance. That is where there's a hemoglobin abnormality. It afflicted King George III. Part of the blood, of course, is hemoglobin in the red blood cells. In this case, the ring, the porphyrin ring that holds the oxygen, is not broken down and metabolized by the cells, so it builds up and attacks the nervous system. So it causes mental problems and blood urine and other symptoms. And here too, you can see that it doesn't skip a generation, dominant inheritance. There are a lot of traits with recessive inheritance, hitchhiker's thumb. For any trait with recessive inheritance, so for recessive traits to be expressed, both recessive alleles must be present. With a dominant trait, only one dominant allele needs to be present. Interestingly, a counterclockwise whorl of the hairline is a recessive trait. Albinism is autosomal recessive also. So that is an absence of pigment in the skin. So sometimes generations are skipped. In this case, this whole generation was skipped, which means that it's more likely to be a recessive trait. Incomplete dominance is such that the dominant allele does not completely mask the recessive one. So one question could be, well, why does a dominant allele, why does it get expressed and not the recessive one? Well, that's a very good question. The terminology that's always used is that the dominant allele masks the recessive allele. I don't know how. There might be lots of different ways in which that occurs, but it turns out that the dominant allele is the one that is transcribed during transcription and the making of proteins and enzymes. So it occurs at that level. The dominant allele just has greater clout. So in this case, these flowers were red and white, but the F1 generation showed pink flowers. And the F2 generation showed that half the individuals were pink. And that's what you would expect because the red allele is not completely dominant. It confers a pink color. So dominant and recessive alleles don't interact. They produce different proteins. And dominant alleles are not necessarily more common than recessive alleles. And most genes exist in more than 2 allelic forms. For example, the ABO blood group in humans. This is a case of multiple alleles. Multiple alleles. And in blood genetics, the A allele is inscribed as an I. They're all I's. I can't remember what the I stands for, but IA is A type, capital A. I capital B is B type blood. I capital A and B equals AB. And little I equals O. And it means the kinds of proteins that are on the red blood cell, they identify you as who you are so that your immune system doesn't attack your red blood cells. So it's a way of self-identifying your cells, your red blood cells. So this shows the kinds of proteins. These are supposed to be proteins. These are the A-type proteins. These are the B-type proteins. This individual has a combination of both A and B-type proteins. And this one doesn't have any. Where'd they go? Just didn't get them. And that's because this individual inherited, I'll just put this in larger writing, both little I alleles. This individual inherited 1A and 1B. For the B type, an individual can be IB, or IB and little I. A type A can be both dominant as well, or IA, little I. So ABO is a blood type that's determined by multiple alleles. Not only that, but in the case of IAIB, there is co-dominance. So the identifiers, the identifying proteins on the surface of the blood cell, they're pretty much equal amounts. This is more complicated, but it's exactly what I just showed. Pleiotropy is when a gene, one gene, has multiple effects. A good example of that is the sickle cell allele. So sickle cell is a pretty interesting phenomenon. It's not pleasant, of course, it's horrid. Sickle cell is recessive. So the small s there indicates the sickle cell allele, which is just one amino acid in hemoglobin is different. So in sickle cell, the protein that is coded for here is hemoglobin. In one amino acid. Big S is normal hemoglobin. So with sickle cell anemia, though, there are a lot of other traits that occur. Anemia can occur, infections, weakness, impaired growth, and even a liver and spleen failure if it's very severe, in particular if an individual is homozygous recessive, and fatality. So a lot of traits associated with sickle cell anemia. So if an individual is SS, then there will be no sickle cell at all. If an individual is recessive, it'll be sickle cell with severe symptoms. But interestingly, if an individual is heterozygous, so these are all genotypes. Sorry, I put that in the wrong place. Those are genotypes. So what happens with sickle cell is that instead of having nice round blood cells, an individual has sickle-shaped, kind of squish-looking cells. But it is not completely dominant. So if you're heterozygous, there are some nice round red blood cells, but there are also some of the smaller sickle cells. And the reason is that the hemoglobin, because it's a different shape, because of that one amino acid, it clumps and it gets sticky and it causes the red blood cell to collapse, basically. But interestingly, sickle cell is still quite prevalent, even though if someone is homozygous recessive, fatality can very well occur before reproduction. And with diseases that cause mortality before reproduction, you would expect that that allele would not carry on into future generations. Eventually, it would be gone. But that doesn't happen with sickle cell anemia because being heterozygous has an advantage. And the advantage is that remember we talked about protists and plasmodium is a malarial protist. So the advantage is malaria is less prevalent in the heterozygous individuals. Yeah. Some traits are determined by 2 or more genes. So we've already looked at one, blood type. What's another one? Eye color is another one. In epistasis, a gene at one locus alters the phenotypic expression of a gene at a second locus. So here's an example of epistasis. We've got mice. They're both heterozygous. They're both heterozygous for 2 traits. And color is one of them. And the other one is unknown at this point. But you would expect to find the 9:3:3:1 phenotypic ratio in your F2 generation, but you don't have that. You have 1, 2, 3, 4, 5, 6, 7, 8, 9. You have the 9, and you've got 3 for brown, but then you've got 4 for white color. You wouldn't expect to find that, 4/16. But what's happened here is that this individual is homozygous recessive for this other allele C. And so that overshadows the color choice or the color outcome. Lots of human characters vary along a continuum. Those are called, no, they're not called, they're called, sorry, they're quantitative, like human skin, hair, and eye color. Eye color is somewhat quantitative too. So here we're looking at, say, skin color, light on this end of the spectrum and dark on this end of the spectrum. And there are 3 alleles that account for skin color. And interestingly, if you're at the light end, you have all recessive. If you're at the dark end, you have all dominant. And if you're in the middle, here, you have all heterozygous alleles. So it's a continuum. You can even plot it. An eye color, I don't really like to talk about eye color too much. It is a continuum, but it's quite complicated. I think there are genes on 3 different chromosomes now. I think that's what the latest information on eye color. I'm not really exactly sure how it all works with eye color anymore. So, excuse me, is everything dependent on inheriting genes? No. The nature versus nurture question, what is the environmental impact? So this isn't very simple. when characters depend on the environment. So a multifactorial character is influenced by both genes and environment. Like, for example, this hydrangea, it's purple, sometimes they're pink, exactly the same genes, but it's something in the soil that makes them a different color. Yeah. So a phenotype includes physical appearance, but our genes don't only have to do with physical appearance. It also has to do with our internal anatomy, our physiology, and our behavior. And that reflects the overall genotypes that we have and the organism's unique environmental history. Yeah, but even when inheritance is complex, the fundamental laws of segregation and independent assortment will apply. Pedigree analysis is where we can look at pedigrees and try and figure out what the genotypes are. So I did have a pedigree exercise. I don't know if we'll do that, but as long as you can understand pedigrees, we've looked at a whole bunch already. These are just some more, but these pedigrees show with the individuals that express the trait. In this case, since this one has a genotype that's heterozygous, it must be a dominant allele. Yeah. So what time is it now? 4:39. I'm just going to pause for a moment. I'd just like to take a quick look at genetic disorders. They may be recessive or dominant. But of course, the recessive ones will only turn up if the individual is homozygous for the recessive allele. When it comes to talking about genetics and diseases, we talk about someone who is heterozygous as a carrier because they carry the recessive allele but are phenotypically normal. Matings between close relatives. They can increase the probability of the appearance of a genetic disease. It's called consanguineous matings. That's the word. That's why I can never remember it. Consanguineous matings. So somewhere in Italy, where was it? Paloma? There were a lot of intrafamilial marriages. And at some point, you had to get permission from the Pope to marry in order that there wasn't as much intrafamily mating. Some diseases, of course, are genetic. Huntington's disease. It's a degenerative disease of the nervous system, but it cannot be detected until an individual is about 30 to 40 years of age. So this individual here is using pedigree analysis of the history of the individual and the phenotypic traits portrayed in the history of the individual to determine the probability of the offspring having Huntington's disease, because by the time an individual reaches 35 to 40 years of age, they may not be having children anymore. So they wouldn't know. Nowadays, it's more likely that your DNA will be sequenced. Of course, many have genetic and environmental components. So some diseases may occur if there's a predisposition, but won't occur unless environmental factors enter the equation. Things like smoking, of course, obesity, they can both contribute to heart disease and cancer. So genetic counselors provide information to prospective parents that may be concerned about family history for a specific disease. This is where probability comes in. So a genetic counselor uses probability to determine what the odds are, or the probability that their children will have the genetic disorder. So it's very important to understand the principles of probability in that case. Another way to determine if a genetic disorder exists may be through amniocentesis. Uh, uh, Amniotic fluid is the liquid that bathes the fetus. Some of it is removed and tested. Another way is chorionic villus sampling. That's sampling the placenta itself. There's many tests available that identify carriers and can help define the probabilities more accurately. And what I'd like to do, I won't do it today, but another day I'd like to show you a video on personal DNA testing. So that's very common now. It used to be very expensive. Now I think it's $250, and you can be tested for your probability of getting certain conditions or disorders or really any kind of trait. But they're only as good as the samples that the particular company has tested. I won't go into fetal testing. Yeah, so let's stop there.